Hello, my name is Rob Durham,
And I'm John Chappell.
and you're listening to the Gyrolab Spin. On today's podcast, we're going to talk about anti-drug antibodies, and we're fortunate to have some material that was put together by our colleague Chris Fox, who organized a workshop for anti-drug antibodies at the Gyrolab seminar. So John, give us an overview of what we're going to hear about today.
Yeah, so we're going to kind of give a brief introduction into anti-drug antibody assays and talk about in particular how the Gyrolab can be used to actually measure anti-drug antibodies. And in particular, we're going to go through a comprehensive development guide giving an overview of experiments that we set out how to develop an assay very quickly on the Gyrolab. And hopefully we'll be able to talk to you directly, and we can share our knowledge and help you develop these assays.
Yeah, this is one of a more challenging area from an assay development standpoint, regardless of the platform that you're working on. There's many things that you need to optimize. So let's get right into it. So we'll start by giving a little bit of background on what exactly an anti-drug antibody or immunogenicity response is.
So what is ADA? So as Rob just mentioned, ADA is an anti-drug antibody assessment or an immunogenicity assessment. And immunogenicity assessment is an important part of biopharmaceutical drug development. And it's an expectation from the regulators, including the FDA and European authorities and other national authorities, to assess immunogenicity, both preclinically and clinically. Obviously, a preclinical response is normally expected when you put a human protein into an animal, but obviously you need to correlate the immune response with any changes in the PK that you're going to see when the drug is dosed. And it's obviously, from a clinical perspective, the main concern is potential safety concerns, and including adverse events, including like induction of autoimmunity, as well as inflammation reactions, etc. But probably the most important thing, particularly from a monoclonal antibody, is potential an antibody can reduce the efficacy of the drug, and it can change the PK, increase the half-life of the drug. So it's very important to assess immunogenicity as part of drug development.
Right, and I think from, especially in the preclinical space, the Gyrolab has some advantages, both in terms of workflow, but importantly in terms of sample volume requirements. So with the low sample volume requirement, you can essentially have the capability to do in the same small sample several assessments.
Yeah, and I think one of the problems with preclinicals, because you're normally using much higher concentrations of drug and dosage, so therefore drug tolerance is a much bigger issue. And I have particular examples where I'm trying to measure 100 or low nanogram per mil sensitivity of antibody with mg per mil of protein there interfering. So you do need to add sort of pre-treatment methods such as acid dissociation. As we're going to go through today, it is possible to fully automate acid dissociation as part of the Gyrolab workflow. So as mentioned, the implication of immunogenicity can cause adverse events such as allergy, immune complex toxicity, even can induce autoimmunity where you start attacking your own body. But probably the main concern, particularly for a lot of biopharmaceuticals, is the impact on efficacy. So you can reduce the PK profile of the drug, so the half-life gets reduced, so there's less drug. And in some cases, the immune complex will continue to circulate. So actually, the drug will actually remain in the body longer. So it can have a big impact on the drug. So it's important to monitor it and to correlate the immune response with any clinical sort of safety issues.
Yeah, and certainly the clinical endpoint as well, right? Even if the drug is held in circulation longer, it may be blocked in such a way as to no longer have its effect, but even though it's hanging around in the body a little longer.
Yeah, I mean, just because you have an immune response doesn't mean that's going to have a clinical impact. So it's important as part of drug development to actually correlate any clinical sort of, the formation of antibodies against events and reduction of efficacy. So it's important to monitor it. When a drug is actually licensed and they actually normally include the antibody incidence as part of the label of the drug. And the immunogenicity instance will actually depend on the type of molecule it is. For molecules that have more mouse sequences, such as chimeric, they're likely to have a higher antibody response. But also you need to think about the assay that was used at the time that the drug was actually released. I'll give a good example. Infliximab, for example, 15%, quite a high immunogenicity rate. But if you actually use quite modern assays, which I've done for like biosimilar programs, for example, you get a much higher antibody incidences than 15% here. That's reported here. You can get 30–40%. But if then and that's particularly in rheumatoid arthritis, but if you look at the same drug in lymphoma, NHL, for example, the antibody incidence is a lot lower because the drug is being used in immunocompromised patients. So you need to. It's important to look at the antibody incidence, but it will change over time as analytical methods get better.
Yep, and depending on as you said, it's really dependent on the particular patient populations that's being exposed to the drug as well.
Exactly. There are many guidances out there to kind of to indicate how to validate and develop these assays. Probably the first paper was the paper by Myers-Sluice, which really gave a good approach for how to develop assays and an indication of assay sensitivity, etc. And this was built on by the Shankar paper, which came later, which gives some improved statistical approaches. And basically these industry white papers effectively have been incorporated into both EMEA, European guidances, as well as FDA guidances. So probably the two white papers are a really good approach to actually validate and develop these assays.
Yeah, and certainly our guidance is based on the theory that was put forward by these papers.
Exactly. But one thing that's probably changed from the early paper is that the sensitivity requirements for clinical assays is actually got lower. So now it's the expectation that you need to be able to measure 100 nanograms per mil of antibody in the presence of drug. So that's an important. So the assays have had to become more sensitive to deal with these changes in the regulations and the expectations.
Yeah, and I know in some of the sessions that I've been viewed at meetings, you see discussions about whether the sensitivity really buys you an improvement in measuring a clinically relevant immunogenic response.
There's a lot of debate in this area, to be honest. But it gives us something as analytical scientists to shoot for. But you know, and at the end of the day, most of the assays that we develop can normally measure 100 nanograms per mil. But this level all depends on the positive control you use. Yeah. If you use different positive controls, you'll get different sensitivity values. So it's important to select the positive control which is the most relevant for clinical.
Right. Oftentimes you'll see somebody using a monoclonal positive control, which is not the same polyclonal response that you're going to get in the clinical population.
No. But monoclonal, from a reagent management perspective, is easier to use and include in over a long period.
Absolutely.
So immunogenicity, very much going back to the early white papers, follows a tiered approach where you try and develop a screening assay. The screening assay is based on a false positive rate. So you want to ensure that you have false positives and not false negatives, because you don't want to miss antibodies if they're there. So the statistical approach is to give at least 5 to 10% false positives in your population. A screening assay, if a sample is positive in the screening assay, then you have a confirmatory tier, and that normally involves adding additional drug. and then if it's a specific response, it will have a signal inhibition in the presence of drug. So you validate that normally alongside the screening assay. But when it comes to sample analysis, you do a screening assay, then you confirm if it's positive. Then you may do titer, so you may dilute the sample to show the strength of the response, or you may do a neutralizing assay, which is normally a functional assay. But it will all depend what neutralizing assay to use will depend on the mechanism of action of the drug. And if it's in general, cell-based assays tend to be the gold standard, but if your mechanism of action is binding, then you can probably justify a competition ligand binding assay approach instead.
Certainly, as we've indicated with these stars, the two places where the Gyrolab is prominently used in these types of assays is under screening assays and confirmatory assays. But depending on the mechanism of action, you could actually use the gyro system for neutralizing assay.
Yeah, absolutely. And also titering as well. You're obviously going to titer your samples using the same method. So
yeah, and then oftentimes the characterization of isotyping, and I know that the folks at Pfizer recently published a paper where they were actually measuring the affinity of the anti-drug antibody as well. So lots of follow-up on those positives.
Yeah, I mean, I've done lots of work in the past trying to measure IgE, for example, which can be extremely hard to measure. So when it comes to the testing outcomes, really, we can have a true positive, which the ADA is present in a sample and results in a positive. We can have a true negative, which means that there's no ADA present in the sample. We can have a false positive, which is, as I mentioned in the screening assay, we're designing the assay to give at least 5 to 10% false positives, which is a good thing. What we're trying to avoid is a false negative, where we're not able to measure the ADA. This is obviously we want to avoid, but obviously if the drug levels are extremely high, there is always a possibility that we can't measure the ADA because of the presence of drug, which will interfere.
And that's why drug tolerance is so critical to really understand where you're at from a drug tolerance perspective.
Exactly, but the trouble is that drug tolerance, like sensitivity, is completely dependent on what positive control you use. So, so it makes it complicated. But obviously, the safety of test subjects is the priority, and we want to we are aiming for false positive, avoiding false negatives. And as I mentioned, we're shooting for a sensitivity around 100 nanograms, and this is based on data that sort of level can have clinical relevance, but it all depends on the patient and the drug itself. But it's a level that we're now aiming for.
Yeah, it's become sort of the rule of thumb from the FDA, and so that's what everybody follows.
Exactly. So looking at the screening assay format on the Gyrolab in more detail, we normally recommend a bridging format, a homogeneous bridging format. The advantage of the bridging format is it allows you to use a positive control from a different species, for example, a rabbit polyclonal, goat polyclonal, and you can use the same assay because any antibody will bind the drug. You can also allows you to measure different isotypes as well, IgM, IgG, etc. All can be detected in the same assay. As I've already mentioned, the positive control is not representative of the population, but it's better to pick a polyclonal when you can because it's more representative of your samples. I've been involved in many programs where we've specifically raised a polyclonal in rabbits, for example, for the study. But in a lot of cases, you can buy your positive control commercially. So you need to find the best source that you can get. And with immunogenicity assays, you're not doing standard curves. You're normally doing a statistical cut point. And as I mentioned, this is set around a 5% false positive rate to try and avoid getting false negatives.
Yeah, and so on that sort of life cycle management of your assay issue, if you've made a rabbit polyclonal, that's going to be a fairly limited supply of positive control. I know some folks have gone to the measure of having them made in goats just to get a larger amount to manage the drug. But if you think about the whole drug development process running into 5 to 10 years, you're going to need to manage that assay for quite a long time.
Yeah, and when you change your positive control, even when it's the same rabbit, if you do a rebleed, a redose, or and take a new bleed of that animal, that response can be different. So
I guess that's one of the reasons why you might use a monoclonal to sort of scale your assay to make sure that your reagents are performing the same every time. It may not be relevant from a population standpoint, but what it can do is say, is my assay still performing year in and year out the way it was?
Yeah, I mean, I've been involved in many validation programs where we've actually validated the assay using both monoclonal and polyclonal. We use the polyclonal for sensitivity and drug tolerance, and then we use the monoclonal for long-term system suitability, effectively, to show the assay performance.
Yeah, that's the thing I'd want to be sure about, is that year in and year out, my assay is still the same.
So when it comes to the screening assay, it's always a balance between achieving the sensitivity that you want, which is 100 nanograms per mil, as I've mentioned in neat matrix, and also the drug actual levels. So the drug will always interfere in the assay, so we're trying to mitigate the level of drug and still trying to achieve the sensitivity at 100 nanograms per mil. In some cases, we may have a soluble target as well, so those also may change, and also in the presence of drug, the levels of target can increase dramatically. So you need to factor in the drug, which can cause a false positive, a target might create a false negative. So you need to, again, it's important to understand the levels you're expecting and include those in your validation of your method. So when it comes to the confirmatory assay, we normally use exactly the same assay as the screening assay. So the main difference is that we normally incorporate the addition of free drug as an assay step. So the free drug is normally either added to the diluent that you dilute your samples as part of your MRD, or you include it as part of your master mix, so that it's there to interfere. And if you've got a specific response, you'll get an inhibition of the signal. So you normally, when you do your cut point analysis, you normally do screening and confirmatory together. Because you've already had the screening assay with a 5 to 10% false positive rate, you use a lower false positive rate for your confirmatory. You normally use 0.1% or 0.1% false positive rate because you have this extra security. And when you do your confirmatory, you also add with and without drug, so you can see that the sample still screens positive as well. Because there is a lot of difficulty when the sample is just above the screening cut point. You can actually, when you reanalyze it, it can then screen a negative, for example.
Right. I mean, it's the nature of a semi-quantitative assay, right?
Yeah.
The other thing that's interesting here is that built into the Gyros system is the ability to easily create the spiked and unspiked samples in your assay setup. So I think that's a real nice feature in the manager that allows you to set your assays up quite. I mean,
the software allows you to do both your screening and confirmatory data analysis as well. So that's another important feature of the software.
Yeah.
So in terms of setting the cut point, you very much follow the industry white papers. Shankar gives a very good statistical approach. We normally recommend a normalized floating cut point for screening. So what this does is actually tie your response values to the negative control on the day of the assay, and you normally do a multiplication or additional factor to set your per plate cut point. And you define this normally in a normal population, or for example, if you go into specific disease populations, you also need to assess whether that disease population has a different sort of cut point factor as your normal population. So your cut point can actually change throughout your study. You can also do an in-study cut point where you use your pre-dose samples to assess whether you get the same false positive rate as you did when you did your validation of the method.
Interesting. And I know that different disease populations is really critical, because when you have an inflammatory disease like rheumatoid arthritis, the patients are spitting out huge amounts of IgGs all the time, and so it can really create a
Yeah, and
high
the other thing
backdrop.
I've seen as well is disease in the disease population, the target concentration is much higher than you've seen during your validation, so that can impact as well.
So what we've got here is an example of what's going on inside the mixing CD. This provides a really nice workflow for automating the acid dissociation to help with that drug tolerance issue that we were discussing. And so let's take a look at this little animation. So here you can see our mixing CD, and it's got the same 15 nanoliter affinity capture column, but it's connected to some extra plumbing. You can see there's a 200 nanoliter sample volume definition chamber up here in this U shape. We've added our sample and then defined it and added it to a mixing chamber. Now the thing that makes this work is that we've got a really strong hydrophobic barrier at the base of that mixing chamber that allow us to add sequential samples into the mixing chamber. And now you're seeing an acid step being added to the mixing chamber. So we've got our sample with the complex drugs to the antibodies have been complex to the drug, and now when we add our acid, we can incubate that for a period of time, and there's a lot of flexibility in this method. You can adjust the timing of your incubation time as well as the mix time. It rocks it back and forth and mixes it a little bit inside the mixing chamber. And now we're adding our master mix with the labeled capture and detection reagents in a neutralizing solution that will bring our pH back to normal pH and allow for a reassociation to occur. And so we're picking a master mix concentration that will compete with the drug that's present in the sample, and then create complexes, and only those complexes that form the bridged by our anti-drug antibody will bind to the column. And then, as we do in our typical assay, we run a laser, or I'm sorry, here you're seeing some washing step. So we'll do washing steps through the bypass channel. And then the last step of the assay then is to run the laser over the CD and scan those 96 well, or those microstructures, those affinity capture columns, and then we'll get our standard three-dimensional retained fluorescence image on the column that will then be the basis for the analysis.
Just a couple of comments here. Automating the acid dissociation can have lots of advantages, because one of the problems I've seen with running plate-based manual assays is that timing of acid and neutralization addition can have a massive impact on the quantification. So, and I've seen massive analyst to analyst differences with the way they actually do the acidification and neutralization. Mm-hmm. So this takes away that kind of variability, because obviously the assay is going to be formed exactly the same way every time, and the timing's always the same. I think another big thing is that our column, streptavidin column, has quite a high binding capacity. So this gives a bit of flexibility in terms of master mix concentrations. We can do much higher master mix concentrations than is actually physically possible even on a high binding streptavidin plate. So there's lots of good ways to improve drug tolerance with the Gyrolab format.
Yeah, and that's the key thing to remember, is that we're competing with the free drug in the sample. And so the higher your master mix concentration, the more you're able to outcompete that. And of course, you're going to pick an MRD for your samples as well. Exactly. It usually runs in the neighborhood of 10% or 20% to do that as well. So now we'll turn to some of the details involved in developing an ADA assay on the Gyrolab platform.
As mentioned earlier, we do have a Gyrolab assay protocol for developing ADA assays. This is actually published on the Gyros user zone, and the protocol outlines the approach for developing an assay using acid dissociation, but also gives an alternative overnight incubation protocol, which can be used on our normal CDs, such as the 200 nanoliter, 1000 nanoliter, etc. but it does provide a structured approach to developing an ADA assays on the Gyrolab, and we're going to go through this guide in detail. So looking at the Gyrolab ADA assay protocol, it gives us a description of the principles of ADA. It gives us an indication of recommendations for labeling, including things like incorporation ratio. It provides full details of what material is required and where to source, and a step-by-step for each experiment and how to perform them, and an indication of what results can be expected from each experiment. Included with the ADA assay protocol are run designs. So we've already pre-populated run designs to give the end user some help setting up the experiments. The protocol very much does emphasize the benefits of using the mixing CD, but as I mentioned in the previous slide, there is an option to do an overnight incubation and also use our standard CDs as well. But obviously, fully automating the acid dissociation very much gives us the best advantage for the ADA assay. So obviously, we are very keen for people to run this protocol.
Yeah, I think the other thing that I would just point out here is for folks that are using an older version of our software, these run designs are designed with the manager in mind. So if you're using an older version of software, you're not familiar with our software, these run designs are specifically targeted for folks that are using manager. Now, you can certainly use the protocol and set up the designs using the old Gyrolab control as well, and we can certainly provide some guidance on that. That's what I wanted to point out here in terms of what the nomenclature is. When we talk run design, that's referring to a run setup that's done in manager.
Yeah. And then just a little note on the end that we found that for some ADA assays that it's better to add the drug in the MRD diluent than it is in the master mix. But we do know that some customers prefer to add the drug as part of the master mix. So you should very much look at both if you want to add the drug at the master mix stage.
Yep.
So looking at the guide for the experiments that need to be run, this guide is designed to be taken into the lab. It provides a list of materials needed for each assessment and gives us structured, stepwise instructions to prioritize actions. So in the experiment guide, it actually gives you an indication of how to set the experiment up. So it gives you a pipetting and dilution scheme, showing you how to prepare your positive controls, but also how to prepare your drug concentrations. So in terms of the development guide, we've actually got 4 structured experiments. So experiment 1 is an initial screen, and then experiment 2 is an assay optimization, followed by a cut point determination in experiment 3, and finally an assay characterization. And what we will do next is go through each experiment and give you a kind of a brief overview of what the aim of each experiment and also an indication of what sort of results you can expect. So the first experiment is a master mix concentration screen. So we normally recommend that you use 3 micrograms per mil and 8 microgram per mil in the presence of drug, and then we add the lower master mix concentration without drug as well. And the aim of this experiment is to look at your an early indication of drug tolerance and sensitivity in the presence of with and without drug, because in my experience you can develop a great assay, very sensitive assay, but you don't know how it's going to perform until it's in the presence of drug. So there's no point developing a really sensitive assay. As soon as you add drug, it wipes out sensitivity. So you need to factor in that drug's going to be present in the sample.
And I don't know, I'm a little bit ambivalent about what level of drug you use. I mean, initially you may want to say, okay, well, my trough levels of drug in the study are going to be X.
Yes.
And so I want to use X as the thing that will challenge my my assay. But sometimes you just wipe everything out and you don't see anything. So I think it's helpful sometimes to maybe pick a couple of intermediate dilutions of the drug as well. So
Yeah,
you're not
I would
gonna,
normally suggest screening a couple of concentrations of drug.
Mhm.
And I know there's a whole debate in the industry of what drug level to actually choose. I think traditionally people have gone very high, so at least 100 microgram per mil, but I think there is some discussion that people should look at slightly lower concentrations of drug. And also it is important that the drug doesn't in some way inhibit the blank signal as well. So
Yeah.
you need to make sure that drug isn't impacting the assay signal as well.
So this just shows then what the experiment layout will look like when you print out your plate map and you get your sheet, and you can see then, of course, whether you're using the mixing CD 96, where we have automated the addition of the master mix and neutralization buffers, versus doing it on a plate where you're going to incorporate the master mix into your dilution of your samples, your positive control, when you're on the plate.
So here is some example data that we generated in-house. This is for an existing biological drug, ERBITUX. And we've looked at the 3 and the 8 microgram per mil master mix concentrations in the presence of drug. And you can actually see that the limit of detection can be calculated in the software. So this is where you extrapolate the curve to 0. And you can see that the 2 master mix concentrations are given very similar sensitivity levels. You can see that the response is higher with the 8 microgram per mil. But based on this data, we will probably continue with this development using the 3 microgram per mil and look at concentrations around that level. And then when we compare the 3 microgram per mil with and without drug, as you can see here that the drug hasn't had a massive impact on the assay signal. And this is this means that we're going to get a good drug tolerant assay, which is very promising for further assay development. And then, as I mentioned, we also want to make sure there's no blank signal inhibition. So sometimes when you add the drug to the blank, it causes a reduction in the signal, even when there's no drug present. So we just want to make sure that there is no signal inhibition, and in this case, there's only a very minor, if any, inhibition in the signal. So we are good to go with the screening confirmatory approach. So based on the data of experiment 1, we then move on to experiment 2, which is a master mix further optimization. So we select the best master mix concentration from the first experiment, and then we expand on that and look at low, medium, and high. So in this case, I believe we chose 1, 2, and 3 microgram per mil. And we're doing all this in the presence of drug because we want sensitivity in the presence of drug. But in this experiment, we also include some blanks to give us an indication of our limit of detection, but also to give us to start to think about the cut point experiment, which would be the next experiment 3.
So here you can see the plate map layout that is produced by the instrument when you set these experiments up. And on the left you have the mixing CD 96, where you have your 1 titration of the positive control that's going to be your standard in this case. And then we have the different acidic buffer that's being used. In this case it's glycine. And then we have high, mid, and low master mix concentrations that will get added in after that acidic buffer, and then of course the wash solutions. On the right you can see what the layout looks like for the bioassay CD, and here, because you're pre-incubating, your master mix is included with each curve, and you'll have 3 curves since you have 3 different conditions of master mix.
Now looking at the results, this is results from a different drug, REMSIMA, which is a biosimilar of Remicade or infliximab. And in this case, we've looked at the 3 master mix concentrations, 1, 2, and 3, in the presence of drug. And then we look at the relative response values and the standard curve, mini standard curve response. and we're looking at the limit of detection in the presence of drug. And as you can see here, that the middle curve, the green 2 microgram per mil, has the best sensitivity, around 47 nanograms per mil. So now we've optimized our master mix and assay conditions, we now need to assess the potential cut point, which is the main part of both the screening and confirmatory assays. So normally this is done with 50 individuals and treatment naive individuals, and we use these individual samples to calculate the cut point factors for screening and confirmatory. Just a note on cut point setting. So the software itself does not calculate a cut point. You actually need to do some statistical analysis outside of the evaluator. Things like a normality test where you look at outliers and also to ensure that you've got a normal distribution of the data. This kind of a better guide for this can be seen in the white papers such as Shankar approaches. We're not going to recommend exact approaches statistically. Please consult with your statisticians and other statistical experts who will give you a best guide of how to handle the data. So in terms of cut point setting, again, it's if you need to assess normality, to actually get normality, you may need to do a log transformation, for example. And in some cases, you may look for non-parametric methods as well. So please, as I mentioned, talk to your statistical experts. We are trying to aim for a 5% false positive rate with the screening assay and a 1% false positive rate with the confirmatory assay. Here we show the data that's generated as part of experiment 3. So in this case, we have 3 CDs of data where we've looked at the response values from all the negative samples, but also we've added negative control and positive control. The overall cut point factor is calculated by looking at the overall data set from all 3 CDs. And then based on the negative control response, you apply a specific plate or CD response, a cut point for each CD. So you enter that factor back into the software. It will draw this line, which will show you visually which samples are above or below this cut point factor. So it's a very good way of presenting the data, and it calculates this for you.
Yeah, it's really nice and clear because you can see which ones of the samples are blue.
Yeah.
Anything that's supposed to be positive is orange, and anything that's supposed to be negative is gray. So I think it's really nice visual for really looking at a lot of screening data. In some cases, you might have as many as 50 individuals here, and so there is quite a bit of data to look at.
Yeah, and it also allows you to assess your false positive rate very quickly. You can see like how many samples are above the cut point. You expect at least 5%, so it's a very good way of presenting the data. So following on now to experiment 4, where we're really looking at assay performance. So we've optimized our assay, we've looked at a preliminary cut point. Now we're going to do a formal assessment of drug tolerance, although in a previous experiment we factored in that we're going to have a certain level of drug tolerance based on the master mix selections, and also we're going to calculate the sensitivity. And what we do use is the cut point factor sort of that we've calculated in experiment 3 and apply them to experiment 4. So this is why experiment 3 is very important for the development of the method. So here are some example results for experiment 4 for the drug tolerance. So for this experiment, we've taken the 100 nanogram per mil positive control, which is what our target sensitivity is in the presence of drug. And we've added increasing amount of drug to see the impact of the drug concentration and when the drug causes the sample to go below the assay cut point. So in this case, we can see the actual assay is tolerant up to 640 microgram per mil of ERBITUX. The good thing with things like ERBITUX and other biosimilars is that there normally are scientific guides out there that the regulators have summarized the performance and indications of the drug. And these can be used to know what your expected concentrations are going to be, the pharmacokinetics. So you know what your assay needs to be clinically relevant. And now for the results of the assay sensitivity. So we take the positive control and we serially dilute it. And we're looking at the level where the positive control sample goes below the cut point. It's probably the best way of doing sensitivity, to be honest. Some people will try and do response curves to actually interpolate the actual sensitivity level, but just finding the first concentration that remains above the cut point is probably the easiest way of doing handling this data. So if you follow most of those experiments, you'll now be at a stage where you have an assay that's pretty much ready for validation. Although you may want to look at other parameters, things like hook effect. So this is when the ADA can cause a negative response because it's so high that it causes inhibition of the signal. You may want to look at selectivity and specificity, where you look at spiking individuals to see if there's any change in the response level in the individuals. That's a kind of indication of matrix effect. You also might want to look at precision and reproducibility of both your negative and your positive controls. But all this is dependent on how you're going to use your assay, what stage your drug development is, and it really is for you to discuss with your regulatory experts and potentially the regulators, and based on local regulations.
Yeah, and again, as always, you want to involve your own biostatisticians to sort of oversee some of the work as well and make sure that you have your final validation in place. But you know what I think we've tried to highlight here is how the Gyrolab can really facilitate this work by automating that assay on the mixing CD. You can improve the way you do your work, as well as the consistency of the performance of an acid dissociation pre-treatment of the samples.
I think another kind of important point as well is that the CDs are very consistent batch to batch. Yep. So you can be very confident that if you use and validate the assay on the Gyrolab, if you over time the assay will perform very consistently.
Yeah, I think one of the things that I've seen that creates the most problems for these types of assays is what's going on with your critical reagents. And I think we've talked a lot at the big meetings, the AAPS and so forth, talking about critical reagent maintenance and characterization and how important that is for the life cycle of your assay. So as with any platform, you know, the quality of the reagents is going to bear quite heavily on the quality of the ultimate results for the assay. Well, we've come to the end of our discussion for today. Thank you so much for listening. This was a little longer form version of our podcast, but I think this is a really important topic. I know that I've had a lot of customers interested in doing anti-drug antibody assays on the platform. So I really appreciate you going through this in great detail, John. I think this will be terrific for our customers and for our team to really understand in detail what Gyros provides in this realm.
Yeah, and if you have any questions, please don't hesitate to contact us. If you want to discuss your specific assay and your specific development, please contact us.
Great, and with that, we'll say until next time.
Thank you much, everyone. Take care.